Hello students. Today our topic is genetic disorder. We start from genetic disorder. Before going to read about the genetic disorder, first here it is given P-degree analysis. P-degree analysis means study of a specific trait in generation after generation, is in various generation, right? How it inherit in next generation. That is, we can study it through P-degree analysis, right? In this case, what is given here? The idea that disorders are inherited has been prevailing in the human society since long, right? This was based on the heritability of certain characteristics features in the family. Certain characteristics features in the family. After the rediscovery of Mendel's work, in 1900, the practice of analyzing inheritance pattern of traits in human being, it began also. Since it is evident that control crosses that can be performed in pea plant or some other organisms, as in Drosophila, are not possible in case of human beings. Right? It is not possible in human beings. Why it is not possible in human beings? Right? Okay. Study of the family history about inheritance of a particular trait provides an alternative. Only we have one manner so that we can study the inheritance of a specific trait, and that is study of the family history, right? Such an analysis of trait, that is called such an analysis of traits, right, in several of generation of a family is called as P-degree analysis, right? So that means we can say study of a trait in several generation of a family is called P-degree analysis. You understand? In the P-degree analysis, the inheritance of a particular trait is represented in the family tree over generations, right? Here, P-degree analysis means what? That is, we represented it by a tree. Tree means what? It is a family tree or P-degree tree, right? Here, what happens? We can understand how the specific trait each inherit in one generation after then another generation, right? In human genetics, P-degree study provides a strong tool which is utilized to trace the inheritance of a specific trait, abnormality or disease. Specific trait, mostly we here study the abnormal traits or disorders or disease. Some of the important standard symbols used in the P-degree analysis has been shown in the figure 4.13, and this figure is given here. Symbol used in the human P-degree analysis. In NCERT, not all the symbols are given, but major symbols are given here, and you can see here that a square, it is represented the male. Square means it represents the male one. A circle represents the female, right? Male, that one, female, circle. Sex unspecified, rectangle, right? It is not a circle. This one is not a circle nor a square, right? Not a square. It is called sex unspecified. Affected individuals. I think it is not going on properly. That here. That one is male square, female means circle, unspecified, it is a rectangle. Affected individuals, they are represented by colored or dark circle, that is affected male. This one is affected male. This one is affected female, and this one is affected, unspecified, right? That is mating, right? Mating, male and female. That one is mating between relatives, consanguineous mating. Consanguineous means what? Blood related. That means what? Blood related mating. Blood related, brother, sister, like, right? And parents above and children below. When we represent this, that is parents, and after that one, these are one daughter and one son is there. In order of birth, left to right, which one is elder? This one is elder, and that side is younger, right? Now, parents with male child affected with disease, that these are normal parents, but the affected son is there. That child means it is a son. Son means because it is a male, so it is a son, it is affected. And 5 means it represents the number of individuals. 5 means it represents the number of individuals. But here, it is not a square and not neither a square nor a circle. That means it verifies the unaffected means unspecified, right? And uninfected, unaffected, unspecified, 5 offsprings. So only these symbols are given in our NCERT, but some more symbols are also here. So first, I think we will talk about the eugenics. Look here, that pedigree analysis is for only human beings. Human beings. First, what is human genetics? Human genetics means it is also called as eugenics, right? Okay, I would describe how eugenics. Eugenics. What is the meaning of eugenics? Eugenics means well-born, and first this eugenics, it is proposed by Sir Francis Galton. So first, Sir Francis Galton is also called as father of human genetics or eugenics. Here, so first, what is here? Human genetics. GENETICS. Human genetics. Human genetics means what? The study of analysis. We can see here. The study of analysis. The study means what? Analysis simply. The analysis of genetic characters and aspects. Mainly they are genetic disorders, right? Or we can say diseases, right? In human, generation after generation. We can say in several generations. That is called human pedigree analysis, and the study or analysis of genetic traits for well-being of human. That is called human genetics. You understand what is there? The study, we can see here, STD, study of genetic traits. Study means what? Analysis, we can say. Genetic traits or disorders or disease, right? In a family. Generation after generation. That means history, simply of genetic traits. Is called pedigree analysis. First thing here, right? It is called pedigree analysis. That is pedigree analysis. But here, one thing is more. When we study these traits for well-being of humans, then it is called as human genetics. To improve the human, we can say here, analysis, that is the difference between the definition. Analysis of genetic traits. Genetic traits or disorder or disease simply, or disease, to improve human, we can say to improve the human, right? That then it is called or termed as, right, human genetics. It is called as human genetics. That is the difference. Improve human means what? Well-being of human, or we can say well-being of human, right? It is called as human genetics. Also, human genetics is also called as human genetics is also called as eugenics. Eugenics, right? Eugenics. And this eugenics, it is first proposed by Sir. Who proposed this one? It is Galton, Sir Francis Galton. So Sir Francis Galton is called as father of genetics. Here we can say eugenics is first in 1883. In which year? 1883. Proposed by Sir Francis Galton. Sir Francis Galton. So, hence, Sir Francis Galton is called, hence, he is called as father of human genetics or eugenics, right? So who is the father of eugenics or human genetics? Is Sir Francis Galton, because he proposed in 1883. First he proposed and give the term eugenics, right? So what is the eugenics? Eugenics means the analysis of genetic traits for improvement of human beings, or we can say for well-being of human. When we analyze the genetic traits only for well-being of human, then it is called as human genetics, right? Or another term for this one is called eugenics. And first it is proposed by Sir Francis Galton. So Sir Francis Galton is called as father of eugenics. You understand? Pedigree analysis means study or analysis of history of a human family to analyze the inheritance of genetic trait, specific genetic trait. When we study the whole study of history, it is called pedigree analysis. And study of history for well-being, that is different. Study of history of a family for well-being of human, that is called human genetics. Do you understand that is what is the difference between pedigree analysis and human genetics? Okay. Now some symbols which Sir Francis Galton or we can say we use in pedigree analysis. Some symbols are also given in our NCERT and some other symbols are also there. So first we have to see the symbols, all the symbols, right, so that we can understand the whole family degree analysis. Because one question will come when it comes, right? We have to understand this is a chromosomal disorder, recessive disorder, dominant disorder. They are autosomal recessive disorder or autosomal dominant disorders. They are sex chromosomes recessive disorder or sex chromosome dominant order. We have to find out this one, right? So first we have to see the symbols, right? Symbols are very important here. What is the symbol? Here I write symbols which are used for P degree analysis, right? And this side, these symbols that is used for first in NCERT it is given square. Square means it is male, normal male. We can say normal, right? Second, if it is given a circle, so circle means it represents the normal female, right? Second, unspecified means not a circle, not a square. It is called unspecified individual. That means in the family we cannot understand that is a okay. They have 3 children. One is boy, one is girl, but third one is girl or boy. There is no idea. Then we use this symbol, right? Unspecified individual. Now second, then after what happens? If then after here that is female and male. Okay, that means what? It is the mating between and human case we can say it is marriage. Marriage especially used for mating, male and female normal mating, right? But another mating is if it is represented by double lines, right? Double line. That means what? It is also we can say marriage, right? But here what has blood related. Blood related. So it is called as consanguineous, right? What is called consanguineous? Consanguineous means blood related, right? Sanguine SGUI, right? GUINEUS. It is not here. Consanguineous. Consanguineous means blood related. Then after what happens? Now marriage it is happens here. And when we represented this one, that is mother, father, and this represent the child's daughter, son, or we can say here this one is daughter, this one is son, and this one is unspecified, right? So this is a means they are mother and this is the father. We can represent it by this one. Means we can say it is a tree, family tree, which is represented here, right? And here, this one represented by means this one is older, that side older, and that side younger, right? That side is younger one. Okay, from we can say left to right, older, younger. Okay. Okay. In this phase, I will tell you. Next one is okay. So these are normal. These are normal. They are in this case no one is affected. Second here also symbol, and they are used for symbol. Which symbol here? If they are, that is, it is colored, right? Then it is called as affected. Affected male. This one represents the affected female, right? Affected unspecified. Individual, right? Affected unspecified individual. Now here, heterozygous and homozygous, they are also, it is also represented by this one, right? Here we can say if it is half colored and half white. Suppose that I draw here this one, that, or that means heterozygous. Heterozygous, two different, right? Heterozygous. Another, if it is given by this one, okay, we will first only circle and a point is present in between. Male, because male is always affected. A male never becomes a carrier. Only female becomes carrier, right? So that one is called carrier. Female. Male never becomes a carrier. Okay, male always affected. Okay. Third is here. Suppose that it is represented by this one and a small dot-like structure is present. So, small dot-like structure, this means what? That represents the stillbirth. Stillbirth, or we can say abortion. Abortion, right? For death, we use another symbol. Suppose that this is given like this one, or and it's crossed like this one. So this one is called death of individual. That is male death. Male is dead, female is dead. So death of individual, right? Death of individual. Another symbol is here for means twins, right? And twins means we know we have two types of twins. Either it is monozygotic twins or it may be dizygotic twins. So when we consider twins, twins are represented by, so normal is represented by this line, right? But twins means represented by, that is mother, father, right? And twins are represented by how? They are represented by simply, okay, here these are twins, right? Twins. If only a, means we can say if in these between a line is also drawn, that means they are called as, look, this, right? Or maybe it is represented by, there is only one option, either they both are male or female. If this line is also present, then this is the symbol of monozygotic twins, right? First mean they are monozygotic females and these are monozygotic males, right? Okay. Second symbol means what happens? Here it is represented by this. Here three options are there, right? Either they are both are male, or one is male and another is female, or it may be both are male, right? This one, it represents the dizygote sequence. Dizygote sequence. So twins are represented by this symbol. Okay. And one more, I think I tell you here, if it is given by like this one, it is present in our NCERT also. A number is given, either this one or this one, or it is look like this one. That means this number, it indicates the number of individuals. Number of son 5, number of daughter 5, number of unspecified child 5. That means it is the number of individuals. Number of individuals. So these are some symbols which we use in pedigree analysis when we form a family chart, family tree. Male, unaffected male means normal female circle, unspecified individual, that is the mating between two different, right? Means we can say here marriage is normal marriage. Here consanguine is consanguineous. Sanguivorous means, already you know, sanguivorous means blood sucking animals. Here sanguine means what? Blood. That is here consanguineous means what? Blood related, right? Marriage is there. Now here it is a, we can say, family tree. It is family tree is represented by this one. And here what is given here? That older is means it is older one is toward the left side, and younger one it is represented toward the right side. So we can understand that means the individual which is, or the son which is leftmost, it is the older one, and the rightmost, it is the younger one, right? Here when they are affected, so males means we have, this one is a colored or a black square is there. Affected female means square, black, or we can say it is colored. Circle is there. Unspecified person, they are affected. Rectangle is there. Now heterozygous is represented by half black and half unfilled. Half filled circle and half unfilled circle of half filled square of, or half filled unsquare. Means this is the heterozygous female, heterozygous male. Here carrier female is represented by a circle and a dot. And I'll tell you that male never becomes a carrier because it has XY, and Y means what? It is the recessive trait, right? That's why here. And here stillbirth, a dot is, if it is represented by a dot-like structure, then it is called aborted or child or stillbirth, right? When they are dead means it is given by a cross, right? And twins, so we have two types of twins, monozygotic, which develop from a single zygote, right? And another one is dizygotic, which develop through two different zygotes. Monozygotes, they are interconnected with a line, and they either both are female or both are male. They never being a one female and one male, right? And in case of dizygotic, we have three conditions. Either they may be both may be male, both may be female, or one is female and one is male, right? But here what happens? In between these two, no line is there, right? No link is there. And if number is given in the square or in the circle or in a rectangle, it represents the number of individuals, number of son or daughters, or number of unspecified individual in the tree. So these are some symbols which are which we have to read for a family, for the study of family degree analysis, right? Pedigree analysis, we can say pedigree analysis. Okay. Here's some information I want to give you: the dominant and the recessive trait in human beings. Human beings means what? Eye color. When we consider the eye color, so eye color means brown is the dominant and blue is the recessive. Blau. Okay. Can I move? Okay. Some traits in humans. Dominant here and recessive. It is recessive, right? Traits. Which traits? First we see eye color. Eye color. Eye color means, okay, how many traits are there? First I write here what are the traits. Eye color, right? Second we have here size is also there. Eye size. Third you know, that is our this ear lobes, right? This is also important. Two types of ear lobes are there. Ear lobe. Fourth, skin color also, you know, skin color is there, right? Hairs are also there. Hairs, hair. Fifth, if we consider the cheeks, cheek, right? Sixth, if we consider which one, we can say one more important, rolling of tongue. Rolling of tongue, right? It can, some persons they roll, but some cannot roll this one, right? RH factor. RH factor, I know you know RH factors, positive or negative. Eighth means skin color. Skin color, right? Sometimes it is also asked PTC. PTC means what? PTC means what? Phenyl thiocarbamate tester. You can see here phenyl thiocarbonate tester. Checker or tester, right? Tester. Okay. Oh, first means eye color. Eye color, we know we have three types of eyes are there: blue, brown and black. Here brown or black color eye is dominant over blue color eyes. That's why this time in the world, blue is means blue eyes are the, means we can say it is the symbol of beauty, right? But blue eyes are very rare, right? Because why rare? Because they are recessive, right? Second, size. If we consider size, large size is dominant over small size. Some eyes are very large, right? Large eyes is also a symbol of beauty, right? Third is eye, that is earlobe. You can see here earlobe. Earlobe either free or fused. You can see here in my ears, it is fused. It is not, because it is not a free. Sometimes they have the ear. You can see this side, right? So this earlobe is fused. But in some person, mostly persons, earlobe is free, right? So here what happens? We can say earlobe free. Free is dominant over fused. You understand? My ears, when you see my ears are fused. It is a recessive trait, right? Free ears means it is a dominant trait. Okay. Now hairs means you understand curly hairs. Curly hairs are dominant over smooth hairs. Smooth means straight hair, we can say right here. STRAIGST. Curly hairs, they are thick, right? You can see here, because capital B or here it is capital B, that is a baldness, right? Gene is responsible. Capital B is present, that means what happens? Baldness is there, confirmedly baldness. Capital B, small b is there, means, as for example in me, you can see I have no hair at that place, and many persons they have no, because they contains capital B, small b. Heterozygous B is there. Heterozygous B means it is sex related, we can say sex influenced characters, right? We can say this one, I will tell you, right? Sex influenced character, because here in male, after means mid age, that mid age means after 45, then what happens? These hairs are gone, right? But in females it has not. That's why it is called as sex influenced. So curly hairs, they are means they represent the dominant traits. Okay, in here. And smooth. Sometimes what have very smooth hair, very shiny and very straight hairs are there. They are recessive. Okay. Now cheeks. Cheeks means in some cheeks there is a dimple is found, right? So when we consider this dimple cheek, it is dominant over simple cheek. Simple, this is the normal. When dimple is formed, in some cases, in some persons, you see this dimple is formed in the cheek, right? So this one is dominant trait. Rolling. Now next one is our rolling of tongue. Rolling of tongue means here roller is dominant over non-rollers. This one is dominant over non-rollers, right? Second is RH factor. So RH factor we know RH positive is dominant over RH negative. RH positive is dominant over RH negative. That's why RH positive 80% of total world population, it is RH positive. Only 20% they are RH negative. And when we consider India, India means it is very high rate. High rate means what? 97% are RH positive and only 3% are RH negative. Okay, PTC are now here. Skin color. Skin color we know. Skin color pigmentation. Melanin skin, we can say normal skin here. Skin with normal or with melanin. Melanin, because it is depend upon the, we know here it is a polygenic inheritance. Polygenic means more than one gene. 3 genes are involved for skin color, right? So normal or melanism, black color skin, is dominant over albinism. Albinism is on white skin, right? And last PTC, taster. So taster means here this one. Who can taste this one? So taster is dominant over non-tasters, right? We can say here taster is dominant over non-tasters. So these are some dominant or recessive traits which are present in human beings, right? So you need to know this one, right? These are important because you are a biology students and you are going to become a doctor. Then you understand which one is a dominant trait or not a dominant trait. So it is important. So you can understand from here which trait is important, which trait is dominant in human and which one is not, which one is recessive. In a normal human, these is present in normal human. These are not a disorders, or we can say these are not a disease, not a disorders, right? But these are traits. These are only traits, and these are autosomal traits. Autosomal traits. As I tell you, when I see this one, why my ears are like this one, why this ears another. So if you have this idea, this one is a recessive trait. That means if it is present in homozygous condition, then it will be appear. If it is present in heterozygous condition, and if it is not a dominant, if it is recessive, then it cannot be present in next generation, right? Okay, now come to the NCERT. What is given in NCERT? Then we will so we now understand the different symbols which are present in, which are used in pedigree analysis. As you have studied in this chapter, each and every feature in any organism is controlled by one or the other gene located on the DNA present in the chromosome. We know chromosome is what? In chromosome, what is present? DNA is present. In DNA, what is present? Genes are present, right? Genes are present, right? And these genes, they are control the traits, specific traits. Specific trait means what? It is a phenotype. Phenotype means what? It is a depend upon the type of genes. Enzymes. Enzymes is protein. Protein simply we understand. Messenger means what? DNA. Simply here we understand that gene controls the specific traits, right? In every generation. DNA is the carrier of genetic information because it contains the genes. It is hence transmitted from one generation to the other without any change or alteration. It is transmitted from one generation to other generation without any change, except in case of mutation, right? However, changes or alteration do take place occasionally. If changes and alteration they take place, then it is called as mutation, right? Such an alteration or change in the genetic material is referred to as mutation. That is given here. A number of disorders in human beings have been found to be associated with the inheritance of changed or altered genes or chromosomes. So, how many disorders are given here? Have to be found to be associated with inheritance of changed or altered genes or chromosomes. Change or altered genes means what? Mutations, or chromosomes. Chromosomal mutation means either through aneuploidy or euploidy, means we can say either through ploidy, trisomy or tetrasomy, or we can say here either through chromosomal aberration or point mutation or frame shift mutation, right? Some Mendelian disorders given here. Mendelian disorders. Broadly, genetic disorder may be grouped into two categories. What two categories are there? First one is called Mendelian disorder and second is called chromosomal disorders. Mendelian disorders means they follow the Mendelian rule, right? Chromosomal disorders means there is some mutation, right? Mendelian disorders are mainly determined by alteration or mutation in the single gene, single gene which is present inside the DNA, we can say inside the chromosome. Means here number of chromosomes are not altered, chromosome aberration is also not there, but a single, which means here we can say it is a point mutation or frame shift mutation may be happened here, right? Here what is given here? You have to underline this one because these are important points. All the statements in NCERT is very important and you have to read this one very carefully, right? Mendelian disorders. Here we have first what is given. Genetic disorder. They are grouped into two categories. First is Mendelian disorder. Second is chromosomal disorder. Mendelian disorders are mainly determined by alteration or mutation in a single gene mutation. These disorders are transmitted to the offspring on the same line as we have studied in the principle of inheritance, right? It may be Mendelian means law of dominance, law of segregation, or right. Third, the pattern of inheritance of such Mendelian or disorder can be traced in the family by pedigree analysis. That is given here by pedigree analysis. Most common and prevalent Mendelian disorders are hemophilia, first, cystic fibrosis, sickle cell anemia, point mutation is example of point mutation, right? Color blindness, phenylketonuria, right? Thalassemia, etc. Right? Some examples are given here and you have to read these examples. Which one? Hemophilia, cystic fibrosis, sickle cell anemia, color blindness, phenylketonuria, thalassemia. These disorders, these are Mendelian disorders which is given in NCERT and you have to read these names. It is important to mention here that such Mendelian disorders may be dominant or recessive. They may be dominant or they may be recessive. But one thing is there, if they are dominant, that means in heterozygous, if they are present in heterozygous condition, so they express themselves. And if they are recessive, that means what? They only express themselves in homozygous condition. When you keep this idea in your brain, in your mind, then you understand this is a recessive trait or this one is a dominant trait, right? By pedigree analysis, one can easily understand whether the trait in question is dominant or recessive. So when we see the pedigree analysis, we understand this is a dominant trait or this is a recessive trait. Either it is present in sex chromosome or it is present in autosomal chromosome, right? Similarly, the trait may also be linked to the sex chromosome, as in case of hemophilia, right? Hemophilia means it is a recessive sex-linked trait. It is evident that X-linked recessive trait shows transmission from carrier female to male progeny. X from female always is it goes to male, because from male, right, to son, when we consider the son, right, so son get X chromosome from mother, not from father, only from mother. That means carrier mother, that if mother is carrier, 50% son, there is a probability or there is a possibility of 50% possibility of affected son, right? If mother is affected, that means 100% sons are affected. Okay. A representative pedigree is shown in figure 4.14 for dominant and recessive traits. Discuss with your teacher and design pedigree for characters linked both autosomes and sex chromosome, right? This is important and you have to practice, right? You have many modules, you have many papers, question papers, previous year question papers, right? So many books are there, MCQs are there. You can get these degree and pedigree, family pedigrees are there given here, family trees are given. Then you have to observe, this one is autosomal recessive, this one is sex-linked recessive, this one is autosomal dominant, or this one is sex-related sex chromosomal dominant trait, right? When you practice, then you very easily understand. Three things you have to hear, right? Observe. One thing. If male and female means if parents are normal and their son or daughter means their offsprings are affected, that means it is mostly autosomal recessive trait, right? Parent is affected, and if mother is parent means father and mother both are unaffected, right? But one son, only son, not daughter. If son is affected, that means mostly the mother will be carrying, right? Sometimes what happens? Father is affected, and sons are normal, but daughters are also normal, right? Then what happens? This X come from father, maybe it is a X-linked trait, X-linked recessive trait, right? If father is affected, mother is normal, and daughter is affected, right? One son is also affected. That means it is X-linked. This means what? It is X-linked trait, X-linked recessive trait. First thing. Second, mother is carrier and father is affected. Mother is carrier. Now we understand that mother is carrier. Heterozygous. Carrier means what? Heterozygous, right? And sometimes what happens? That is, mother is affected, but in second case what happens? Son is unaffected, daughters are unaffected, or anyone is offspring is affected. That means it is autosomal. Means if it is randomly come, that means it is autosomal dominant. These are some specific criteria through which you can understand this is an autosomal recessive, or this one is autosomal dominant, or this is an X-linked recessive, or it is X-linked dominant. Y-linked traits means what? Y-linked traits always they are present in males, father to son, son to. Y-linked trait you easily understand. Like as for example, Y-linked trait that is we can say hypertrichosis. Hypertrichosis means maximum hairs are present in, like in shoulder, in backside, in chest. Hairs are present in male bodies, right? Heterotrichous. So this trait it comes from father to son only, right? So these are some means I tell you some symbols through which we can understand which type of disorder this one. Okay. Here two examples are given, right? Now here you understand through this. When we analyze this one, we don't know this means okay. Here it is given, so understand we know it is a myotonic dystrophy, right? Myotonic dystrophy because Dystrophy, we know dystrophin, that is a gene. Dystrophin, it is the largest gene which contains 2.4 million base pairs, right? This is the largest gene, dystrophin. Due to the disorder of this largest gene, this myotonic dystrophy happens, right? Myotonic muscles degenerates. Here you can see female is affected but male is not. But in this second generation, this is the F1 generation, parents, and this is the F2 generation, second generation. In F1 generation, this is the F1 generation, sorry. In F1 generation, what happens? This is the older one, then they are smaller, right? Okay, this one is normal, this is affected. This one is normal, this is affected. Here we understand parent, father is normal, mother is affected. Mother is affected. Can you tell me it is a sex-linked trait? Here, if it is sex-linked trait, suppose that it is a sex-linked trait, means it is X, X and XY. Then we understand it is sex or non-sex. You understand from here, right? I am only telling for how could we understand that is a autosomal or a sex-linked trait, recessive or dominant. If they are X, if it is X-linked trait, means we can say it is a, because it is affected. So we know if it is a sex-linked trait, means either it is dominant, then it is affected, or it may be recessive. Recessive means in case of recessive means both are affected. Recessive because recessive can express itself in homozygous condition, right? If it is recessive trait, then what happens? It both X contains this one. That means this normal, it never be, that means it, this male, right? This male, what happens? It makes it wrong. If both X contains this trait, right? If it is a recessive trait and if both X linked recessive trait, then both X contain this trait, then means this also it contains X from mother and Y from father. X means it contains this one, that means it should be, but it is normal, that means it is not a recessive trait. So first we understand that it is not a X linked recessive trait, right? Okay, second, it is a autosomal recessive. Can it is a autosomal recessive? Autosomal recessive means simply same. These are autosomes. Autosomes means they are both are XXXY. I write here. They are autosomes, right? AA. I should write here AA. But autosomes means what? Only one. If it is a recessive trait, that means also both X contains this one. Both X contains this one. Both X contains this one. Then how this one is affected? How this one is affected? This one is affected only one case if this is also contains X. But if it contains this one, right? Then what happens? Then how this is affected? If this X contains this one, then how it is affected? If it is a recessive trait, so we have two things. It is affected because it contains both, means both autosomes, it contains the same trait, right? So both are present, so it is affected. We can say it, if father is affected, so it is also affected, we can say correct. But father, it is recessive. Suppose that it is recessive, mother is affected, father is contains recessive trait, that means it is not affected. In this case, what happens? This male is affected. The male means we can say how it is affected, because it get one X from this side and one Y from this side. It cannot be affected, but it is affected. That means it is not a recessive. Second, here what happens? It is affected, but how it affects? Okay, it can be affected, yes, but why it is not affected? That means it is not a autosomal recessive. Okay, one thing only is there, that is autosomal dominant. How it is autosomal dominant structure? Because if it is autosomal dominant, that means only one, and it is normal. Then what happens? If this A comes to that side, because X1 chromosome come to that side, right? Then it is affected. And here it also this X come from mother, then it can be affected. We can say it is a autosomal dominant trait. And what is given here? Autosomal dominant trait, right? Do you understand here? You can see here both are normal, but their sons are affected. Simply I tell you, both are normal. Why they are normal? That means they are. This is a recessive trait. This is a recessive trait. Now Second, it is autosomal or sex-linked recessive trait. Sex-linked means what? XY, X-linked recessive trait, right? So when we see this one, because these are normal, but their son is affected, son or means their son or daughter is affected. If it is X-linked trait, then we understand that X-linked trait means son is affected, but daughter, one X chromosome come from the father, right? It cannot be affected. That means it is an autosomal recessive trait. Autosomal recessive means that it is present here and it also present this, right? So this, when these two traits come to the son, it is affected. When these two traits come to daughter, it is also affected. But when these traits are, look here, these are normal. Normal means one contains this one, but another is normal, right? Sorry, it is normal, but it is 1A and 2, right? Then what affected? It is affected. If it affected means what? It is normal means it may be heterozygous. It may be heterozygous. And here we understand these father are heterozygous for autosomal recessive trait. We understand this one is autosomal recessive trait. For example, sickle cell anemia. So through this, you understand which one is autosomal recessive and autosomal disorders. Color blindness. Here spelling is mistake. You understand this is the color blindness. It is a sex-linked recessive disorder due to the defect of either red or green cone of eye, resulting in the failure of discrimination between red and green color. Red and green color, right? Blue color is also there. This defect is due to the mutation in certain genes present in X chromosomes. It occurs in about 8% of male and about 0.4% in females, right? Females. This is because the genes that lead to red-green color blindness are on X chromosome, and females means they have both X chromosome. Male have only one X chromosome and female have two, right? The son of a woman who carries the gene has a 50% chance of being colorblind because it never becomes a carrier, because it is an X-linked trait. So females, if they are present only one X chromosome, they becomes a carrier. The mother is not herself colorblind, it is only carrier because the gene is recessive. That means that is the effect of suppressed by her matching dominant normal gene, that is matching normal X gene. A daughter will not normally be colorblind, but it is a carrier unless her mother is a carrier and her father is colorblind. Only one case is there, when his mother is carrier and her father is colorblind, then 50% chance, 50% daughter and 50, you can say 50% possibility of a daughter will be colorblind, right? Only 50%. Hemophilia. Second is, it is also a sex-linked recessive disease like to color blindness, which shows its transmission from unaffected carrier female to some of the male progeny has been widely studied. In this disease, a single protein that is a part of cascade protein involved in the clotting of blood is affected. And due to these factors, we have three types of hemophilia. Hemophilia means we can say ABC, simply alpha, beta, gamma, right? That means what? Hemophilia means A means what? That is due to the due to the lack of the factor, due to the defect of the factor 8, factor 9 and factor 11, right? So three types of factors are absent in case of in cascade enzyme, right? So it is this hemophilia, what happens? Blood clotting never happens. Due to the absence of blood clotting, blood continuously goes on, right? Means bleeding cannot be stopped. That's why it is also called as bleeding disease. Due to this, in an affected individual, a simple cut will result in non-stopping bleeding. The heterozygous female that is carrier for hemophilia may transmit the disease to the son. Only son get this one, right? So this means heterozygous female, it is carrier. That means it is heterozygous female means it is phenotypically it is normal, right? But genotypically it is carrier. But if this is a recessive trait, if it is present in son, so it becomes affected. The possibility of female becoming a hemophily is extremely rare because mother of such female has to be at least carrier and the father should be hemophile, unviable in the later stage of the life. The family pedigree of Queen Victoria shows a number of hemophilic descendants as she was the carrier, right, of the disease. Sickle cell anemia, second one is there. Sickle cell anemia, we know it is a autosomal recessive linked trait. This is an autosome linked recessive traits that can be transmitted from parent to the offspring when both the parents are carrier for the gene. When both the parents are carrier, if both parents are carrier, right? Carrier means because it is a recessive genes, so means it is present in both, right? For X-linked trait, that means males never being a carrier, right? That I am saying. For autosomes, they may be carrier because autosome recessive only. For autosome recessive, they may be carrier, right? So autosome is different. XY chromosome different because XY chromosome, what happens in male, XX chromosome is present, but in female, male XY chromosome is there. Y is a recessive chromosome, right? That's why in case of male, what happens, they are affected, but in female, XX both are there, and X means it is dominant over the recessive trait on another X. So the expression of this trait is X-linked trait. It is suppressed. That's why female becomes the carrier. Phenotypically, they are normal, genotypically, they are carrier. But male, they are always affected. But in case of autosomes means both chromosomes are same dominant or same recessive, we can say. They both are same. Then what happens? If it is present in one chromosome, so they are not. If it is recessive and present on one chromosome, so they cannot express. These traits, they cannot express. That means here we can say, in case of autosomes, male may be a carrier, female may be a carrier. In both cases, it is same. That's why it is given that can be transferred from parents of offspring. The disease is controlled when both parents are carrier for the gene or they are heterozygous. The disease is controlled by a single pair of allele HA, HB, S. As for sickle cell anemia, out of three possible genotypes, only homozygous individuals for HBS, homozygous HBS. HBS means hemoglobin. Hemoglobin, it is made up of 4 polypeptide chains, 2 alpha and 2 beta. That beta chain is, right? Heterozygous HB, HBS individuals appears apparently unaffected, means they are phenotypically unaffected, but they are carrier for the disease as there is 50% probability of transmission of the mutant gene of the progeny. 50% means because we know HBA come to the, in case of this one, we have 2 pairs, HBA, 2 genes are there. So one genes go to another gamete and HBS goes to another gametes. So 2 different type of gametes are found. If fertilization occurs through HBS, it is transmitted. Fertilization through HBA and the transmission, 50% of chances is here, right? The defect is caused by a substitution of glutamic acid. Substitution of glutamic acid through valine. Okay, we will read this one. You understand here, a figure is given, this is the normal cell and this means it becomes a sickle cell-like structure. And here, you can see, this one is our DNA. This is our DNA. This side, it is the DNA fragment. This one is our messenger RNA. It is given messenger RNA. That means here what is? Transcription. Transcription. And through translation, a protein is formed. A protein is formed, right? This is translation. This one is transcription. You know GTC. This is the another. This one is not. This strand. This one is coding strand of DNA, and this one is this one. This one is template strand. Template strand. GUG, this is the messenger RNA. Same thing is also there. Coding strand. This is the template strand of DNA. Both are DNA. This is the DNA. And through transcription, messenger RNA GAG is formed. What is the difference here? You can see here T is here A. Means T is replaced by A. Right? This one. And that makes the all difference. Here GAG, here A. A is there. Right? Adenylate acid is there, but here uridylate acid is there. and that makes the whole difference. At GAG, it is responsible for the attachment of glutamic acid, whereas GUG, it is the coat which is responsible for the attachment of valine in transferrin. Right? That's why glutamic acid is replaced by valine. That is replaced. So that is HbA normal peptide, and that is HbS. This is called as point mutation. Point mutation. It shows point mutation. Only a single point mutation. Others are there. Which sixth amino acid is changed, or we can say sixth genetic code is changed. Right? You can write here Val his Luther pro blue. Val his Luther pro Val glue. That valine glutamic acid is replaced by valine, or we can say here in messenger RNA adenylic acid is replaced by uridylic acid. It is a type of substitutional mutation, point mutation. A type point mutation is substitutional. That means here purine is replaced by pyrimidine. Okay. Glue by valine at the 6th position of the beta globin chain of the hemoglobin molecule. Right? The substitution of amino acid in the globin protein results due to the single base substitution at the 6th codon of the beta globin chain for GAG to GUG. That I already tell you. Adenylic acid is replaced by uridylic acid. The mutant hemoglobin molecule undergoes polymerization and under low oxygen tension, causing the change in the shape of RBC from biconcave disc to elongated sickle cell-like structure. Okay. Now, phenylketone. Next disease is phenylketonuria. This inborn error of metabolism is also inherited as the autosomal recessive trait. This one is also a autosomal recessive trait. The affected individuals lack an enzyme that convert the amino acid phenylalanine into tyrosine. As a result of this, phenylalanine is accumulated and converted into phenylpyruvic acid and other derivatives. Accumulation of these in brains results in mental retardation. These are also excreted through the urine because of its poor absorption by kidney. That is called phenylketonuria. Urea is present in urine. Now, thalassemia. So, phenylketonuria, it is a inborn error metabolism, right? And it is also a autosomal recessive trait. Thalassemia. This is also an autosomal linked recessive blood disease transmitted from parent to the offspring when both the parents are unaffected carrier for the gene of heterozygous. One thing you understand here, if it is autosomal linked recessive genes, that means what? If both parents are heterozygous condition, both parents are phenotypically normal, but they are carrier, then the offspring, at least 50% offspring, they will be affected. Either they are male or female. The defect could be due to the either mutation or deletion. Either we can say mutation or deletion, which ultimately results in the reduced rate of synthesis of one of the globin chain. Reduced rate of the synthesis. Reduce means number of amino acids will be reduced here, right? Alpha and beta genes that make up the hemoglobin. That is here two things are there, thalassemia, and when we consider the thalassemia and sickle cell anemia, so sickle cell anemia is a qualitative mutation, but thalassemia it is a quantitative mutation. Because here number of amino acids will be reduced. This causes the formation of an abnormal hemoglobin molecule resulting into the anemia, which is characteristics of the disease. Thalassemia can be classified according to which chain of the hemoglobin molecule is affected. In alpha thalassemia, production of alpha globin chain is affected, while beta thalassemia, production of beta chain is affected, right? Alpha thalassemia is controlled by two closely linked genes HbA1 and HbA2 on chromosome 16. Chromosome 16, right? Both, look here, both chains are formed through two different chromosomes, not through a single chromosome. So, alpha chain, it is formed through 16th chromosome in each parent. It is observed due to the mutation or deletion of one or more of the 4 genes. The more gene affected, the less alpha globin molecule produced. Only reduced is there, no more. while beta thalassemia is controlled by a single gene HBB, B for beta, A for alpha, right, on chromosome 11, 16 and 11, beta for 11, alpha for 16, right, of each parent, and occurs due to the mutation of one or both the genes. Same here, thalassemia differs from sickle cell anemia in that the former form is sickle cell anemia is a quantitative problem of synthesizing of too few globin molecules, while the latter means what? Thalassemia is latter means what? Sickle cell anemia, it is qualitative problem of synthesizing an incorrectly functioning globin. Right? Do you understand here? Thalassemia means reduced genes are there. Sickle cell anemia means number of amino acids is same, but one amino acid replaced. Which is replaced? Glutamic acid is replaced by valine. But in case of thalassemia means number of amino acids are reduced. That's why it is called quantitative abnormality. And sickle cell anemia means qualitative. Now here chromosomal disorders. Many chromosomal disorders means it is due to the aneuploidy. Aneuploidy means number of chromosomes are increased or decreased. Number of chromosomes increased or decreased. Okay, in meiosis what happens? Gamete formation means meiosis is there. In meiosis means prophase first what happens? These chromosomes they exchange, means they are crossing over is there. Then after anaphase is there. In anaphase what happens? The chromosomes they move to other poles, means opposite poles. When they are moving opposite poles, sometime what happens? More chromosomes goes to one pole, another means less chromosome goes to another place, and that makes here aneuploidy, right? The chromosomal disorder on other hand are caused due to absence or excess, that is hypoaneuploidy, or excess means hyperaneuploidy or abnormal arrangement of one or more chromosomes. Failure of segregation of chromatids during cell division results the gain or loss of chromosome called aneuploidy. And you understand, it is due to the prophase, it is due to the anaphase first or due to the anaphase second. Failure, the segregation of chromatids. So chromatids segregate in anaphase second, right? Chromosomes segregate in anaphase first. Results the gain or loss of chromosomes called aneuploidy. For example, Down syndrome result in the gain of extra copy in 21st chromosome. 21st means Down syndrome. Similarly, Turner syndrome is result is due to the loss of X chromosome in 23rd pair, X0. So Turner syndrome is it shows monosomy. Down syndrome it shows trisomy. Failure of cytokinesis after telophase stage of cell division result in an increase of whole set of chromosomes. Set of chromosome is here. Two types of, I tell you here, ploidy means two types of ploidy. It is in mutation and in detail I tell you, right? Ploidy means in two types, either it is euploidy or aneuploidy. Euploidy means it is if the set of chromosome is increased or decreased, means 2n, 3n, 4n, 5n, polyploidy. It is called polyploidy, right? And if the number of chromosome increase or decrease, that is called aneuploidy, right? Mostly in case of animals, aneuploidy is there, and in plants, that is polyploidy is also, right? Mostly, I am not sure every time. Here, failure of cytokinesis means cell division after telophase stage of cell division result in an increase of whole set of chromosome in an organisms, and this phenomenon is known as polyploidy. This condition is often seen in plants. The total number of chromosomes in normal human cell is 46, that means 23 pairs. Out of these, 22 pairs are autosomes and 1 pair is sex chromosomes. Sometimes, too rarely, either an additional copy of chromosome may be included in an individual's, or individual may lack one or any one pair of chromosome, right? This situation are known as trisomy or monosomy of a chromosome, respectively. Trisomy means when extra chromosome is come. Monosomy means when one chromosome is removed. As for example, for trisomy, that is our Klinefelter syndrome, Down syndrome, they represent the trisomy. Monosomy example is Turner syndrome. Such a situation lead to very serious consequence in the individual. Down syndromes, Turner syndromes, Klinefelter syndromes are common example of chromosomal disorder. Down syndrome, the cause of this genetic disorder is the presence of an additional copy of chromosome number on 21st trisomy. It is also called as 21st trisomy. This disorder was first described by Langdon Down in 1866. The affected individual is short-statured with small round head, furrowed tongues, partially open mouth. Every time saliva is released from that mouth. Palm is broad with characteristic palm creases are also there. Physical, psychomotor and mental development is totally retarded. So this shows the EDS, right? Klinefelter syndrome, that is a trisomy of 25. Klinefelter syndrome, it is a trisomy of 23rd pair. This genetic disorder is also caused due to the presence of an additional copy of X chromosome resulting in the karyotype of 47 and it is represented by 44 plus XXY. Such an individual has overall masculine development. However, the feminine development of breast, that is called gynecomastia, is also expressed. Such individuals are sterile male. They are sterile males. They are sterile male, right? Turner syndromes, that is also monosomy of 23rd chromosome. Such a disorder is caused due to the absence of one X chromosome, that is 45 with X0, and it is represented by, this one is represented by 44 plus X0. This one is represented by 44 plus XXY, right? Such females are sterile. Here what happens? Females are sterile, right? Are sterile and ovaries are rudimentary besides other features including the lack of other secondary sexual characters. Okay. So this is the given in this chapter. No other inheritance is also given here. Other characters are also there, right? But we need not to go very outside, right? If you have any doubt, you can ask. Okay. So Down syndrome, Turner syndrome, Edwards syndrome, 13th chromosome trisomy is Edwards syndrome. 5th chromosome, when it is a small arm is defected, it is called as Cry-Du-Chat syndrome, right? Philadelphia syndrome. There are many syndromes are present which is present in different chromosomes, but it is not given in NCERT. So we need not to read this one because these are garbage for us this time, right? Because we are preparing for NEET exam. Okay, if you have any doubt, you can ask, right? Okay, thanks.